Okay, let's keep going. So we were talking about opposition of forces as a regulatory puzzle and how that can be prone to particular perturbations that can lead to disease. And so I had that in my mind, and so there it was. And then several years later, I was talking to the same person that I talked to about infective dose. He's an encyclopedia of infection. He was going on telling me various things about infection. And then he started mentioning something that triggered this idea about opposition of forces. And the issue is that if you think about an infection and you think about an immune response, it's very important for an immune response to recognize an infection and start the defense very quickly. Because when a pathogen gets inside of a host, it starts to replicate and it grows exponentially, which is very fast. And it becomes a race. If the pathogen can grow sufficiently large in its population size before the host immune response turns on, then often severe disease or death follows. On the other hand, if the host can recognize it's being infected and start the immune response and all the defenses, then the host often controls the infection and may not get sick at all, or at least can control the infection. And there's a lot of data showing that this is the case, that it's really a very time-sensitive race in terms of success or failure of infection, and from a host point of view, control of infection and preventing disease. And so that means that the hosts have to trigger an immune response very quickly when there's a signal which suggests that maybe it's infected. And when you have a situation where you have to respond to something that might be happening very quickly, then there tends to be a lot of mistakes, a lot of false positives. So there's a tendency for potential for a lot of false positives in the immune response. And this is a real problem because when you feel sick, when you have an infection, that's mostly your immune response that makes you feel sick. Your immune response is a very powerful set of poisons and weapons. Your body's basically firing off cannons and sending out toxins and doing all sorts of nasty things. And you feel sick, you can sort of think of that as friendly fire or collateral damage because you're turning on all of this tremendous defensive work to try to get rid of an infection, and it really makes you feel pretty bad. And it can be very damaging to your body. Your immune system is actually often causes a lot of damage to your body. And so turning on the immune response when it's not needed is a very bad thing. And so what you have is a situation where you need to detect a signal under very constrained and rapid conditions, which will lead to a lot of false positives. Therefore, you have to have a set of compensatory negative regulators that can turn things off. In effect, say, oh, that was a mistake. Turn that immune system off before you start hurting, your own body. You don't really need it. It wasn't really a dangerous infection. And so it's known that it's been known that there are many negative regulators of immunity. That's sort of a known phenomenon. It's understood why, because immune responses are damaging. But what my friend was telling me was that actually people are seeing actually a lot of negative regulators of immunity are appearing. And I was thinking, but that's kind of puzzling because, you need some negative regulators, but to have these big gene families that are devoted to turning off the immune response, it seems like there's almost as many genes to turn off the immune response as there are genes to turn it on. And that made me think, well, maybe if the immune response is regulated by this opposition of forces, turn on very fast or you're going to die. Turn off because you're making a mistake. And so whether or not an immune response actually develops then it becomes this opposition of regulatory forces. That means that maybe a lot of pathologies associated with immune misregulation come from this opposition of forces. And there are a lot of pathologies with immune misregulation that are known. So then we looked in the literature and we found that there are indeed these large families of negative regulators of immunity in plants, in vertebrates, and many different organisms have these large families of negative regulators, and they evolve very fast. over evolutionary history, which is always an indication that they're under very strong, powerful selective pressure. Now, I think most people, if you would ask them, well, do you think negative regulators of immunity should be under strong selective pressure? Usually the answer would be no, because the negative regulators of immunity are not directly responsible for recognizing attack. It's in the recognition of attack that you get rapid evolutionary response, because pathogens change and the hosts have to recognize changing pathogens. But negative regulators of immunity are internal to our own system. They have nothing to do, usually, you would think, with external recognition. So they shouldn't be something that's very rapidly evolving, but they are. And so um, that's basically the situation. I could refine that a bit more, but I told you that story because this is an example of how in daily life, through conversations or things you hear, you can make connections. So this business about opposition of forces and genomic imprinting and ideas about mothers and fathers and growth rate and all that sort of thing, that was something that that was David Haig's work mostly, somebody else and some other people had developed that work. That was not my work. But just being aware of it and having it in my mind that opposition of forces is very important, then tuned me into um, hearing other things. And so what you have to be able to do is recognize it's something that even if you don't know what to do with it, that's really important. And I don't know what to do with it, but I'm going to hold on to that and kind of keep track of that. Of course, you don't really do that. You just, um, but it makes an impression on you. So what you have to do is get to the point where you're good at getting yourself to make an impression on yourself when you see something that's interesting to hold on to. Um, but these connections are really interesting, and that's really, I think, how you see new things. At least that's been for me. So that was a digression. And what I want to do in the remainder of the time is we're going to skip some of the puzzles because this is just an overview to these first two lectures. And I want to pick out a couple of them that I think pull out some particularly interesting connections. So this isn't so much by way of illustration of what puzzles are, but I want to tell you about are some really powerful unsolved puzzles in the in modern science that I think are really exciting um, aspects of science. So I'll try to do two of them. Maybe we have enough time, maybe we don't. We'll see. But I'll pause briefly just in case you have any questions right now. Do you want to ask anything?
Or I had a question.
Yeah.
You're talking about those large gene families that are negative regulators. Do you know like what kind of proteins they are?
Well, some of them are microRNAs, so they're not proteins at all. That was interesting. But there's other families I don't remember, but we wrote a paper. So if you send me a chat or something, or get in touch with me, I can send you the paper if you're interested in it. I'd love to talk about it. It's
I do
it
too.
hasn't. It's a pretty recent, and not much has been picked up on it. So I think it's still an open problem that really nobody's really followed up on. And but the paper in Plants about the microRNAs was really interesting. There's this huge family of negative regulators of immunity microRNAs in across a wide array of different plant families. I think a really interesting subject.
All right, yeah, I'll send an email with that. That's interesting. Thanks.
Yeah. Okay. Good. Anyone else? Okay. So let me see. I'm still deciding which one to do next. Well, we'll do cancer next. And let's see. So we'll talk about cancer next. And cancer's cancer raises many interesting puzzles. And it never really occurred to me that that was the case for many years. My only feeling about cancer was I didn't want to get it, but I never really thought about it as a subject, as an evolutionary biologist primarily. But then I had a friend who became really interested in cancer research, and he started talking to me about it. And at first I was like, well, yeah, that's fine. I'm not really interested. But then one day I realized that um, it sort of struck me. Cancer is the failure of our normal regulatory controls and protections on our tissues in our body that break down. We have a whole variety of things that protect our cells and tissues from overgrowing. And our body recognizes when something isn't right, and it kills off the tumors in very early stages, and your cells have all sorts of checks and balances and controls. And so I started getting interested, and then I realized that um, one of the most fundamental things in biology is how do we study, because I'm interested in how organisms are designed, how they're put together, how they're built, so to speak, and that biology is really the science of reverse engineering. You have these things that work. They're incredibly complicated, beautiful, well put together things that work, and we don't know how they work. That's a how question. We don't know how they work, and we don't know why they're put together the way they are. And most biological research is reverse engineering. How are these things put together to work? And the analogy I like to use is maybe I've already mentioned this, is imagine that somebody gives you a car, an automobile, and you've never seen one before, and they say, okay, you've got a year, I'm going to leave you alone with this car. And when I come back, I want you to tell me how it works, how it's put together. That's actually a pretty hard question. If you've never seen an automobile before, you can drive it, everything, the steering works, the brakes work, but how? If you open up the hood of the car and you look in the engine, there's thousands of wires and connections and things that are hooked up, and what do they do? And how are you going to figure it out? So one thing you might do is you might reach inside if you see a wire and you could cut that wire. And if you cut the wire and the brakes don't work, then you go, oh, this wire is associated with the brakes. So if I take this wire and I trace it to where it goes, I can find out some module in the car that has to do with the brakes. And if you keep on doing that by breaking things and seeing what broke and what function was disturbed, you can put together a story about how the thing was designed. And that's actually, anybody who studies genetics knows that the way you figure out what a gene does is you knock it out, right? Because if you just look at an individual that works and you say, okay, here's a gene in an individual and the individual is fine, you have no idea, is that gene involved in metabolism? Is it behavior? You can't really tell. But if you knock out that gene and the individual then can't see, then you say, well, the gene has something to do with vision somehow. And so that's really that is the single greatest mechanism of biology is knockouts. And so then I realized that cancer is really a natural set of knockouts. That things are breaking down in your body that normally protect you, that you have these things all built in. And as they get knocked out, you're breaking down the normal protections that design your body, and eventually you get the pathology that follows. And so I realized that because cancer is a set of failures, and failure is how we study design. I like to say that failure reveals design. And once I made that connection, then I became totally fascinated by cancer as a scientific subject, because cancer is actually one of the most insightful subjects. And if you look at the history of modern biology and you trace back, how did we learn about how cells work? How did we learn about DNA repair? A lot of that research started in cancer research. And so it's actually not surprising when you think about it now in retrospect because of this relationship between failure and design. Did you want to ask a question, Thomas? Oh, you're stretching.
I was fixing my light, sorry.
Oh, okay. Yeah, I just see your hand going up. Okay, cool. So anyway, so I became interested in cancer, and so I started looking into various things. And then there became some puzzles, and there are some classic puzzles. And so I'm going to do is start out and show you, and we're going to go really fast through this because I want to lead up to, I'm not going to talk so much about cancer right now, but I want to show you the connections between cancer and some other things, but I'll talk a little bit about cancer. So I need to show you some pictures, so we'll do this. And do that. So these are what are called cancer incidence curves. This is basic epidemiology. So here's age, and you see it's on a log scale. And this is the number of cases per year that develop. And this is a log 10 scale. So 1 means 10 to the 1 or 10. That's 100. That's 1,000. That's the number of cases per year per 100,000 individuals of the population. So this is 1,000, which is 1% of the population getting cancer. So men, when you get to be, about 75, about 1% of men per year will get prostate cancer. Okay, so these are the age incidence curves on a double log plot. And it was known, I don't know when the first really known, at least by the 1940s, that when you plot various types of cancer in different tissues on these double log plots, you often get these very regular curves. So this is pretty much a straight line, and then it levels off. And this is pretty much a straight line, and then it levels off. And many different cancers have this age incidence curve property. And in the 1940s and 50s, people didn't know really knew almost nothing about genetics in cancer. It was just an observation. And they recognized this as a puzzle. They sort of felt that, when you see great regularity, it's sort of like, well, if we could understand why this was so, maybe we would really understand something that's interesting about cancer research, cancer biology, but we don't know what it is. And so in 1969, a guy named Ashley had the very clever idea of thinking, this is sort of like a knockout idea. So this is on the bottom, this is a curve of colorectal cancer in the population as a whole. And you see it's pretty much a straight line on this double log plot. It levels off a little bit later ages. But in this slope is about 4 or 5, I think. 5, I guess. And that's actually very steep because this is on a log plot. So it's really going up very fast with age, but it's very regular. And so why is it so linear and so regular? And so there were various theories that were proposed. And Ashley had the idea that you could really get a lot of insight into this incidence of cancer with regard to underlying causes if you looked at inherited cases and you compared inherited cancers with cancers that are not inherited. And people knew that there were genetic mutations that were inherited in families that predisposed them to cancer. In particular, it was clear that there was a gene that could be mutated in families that would cause a predisposition to colon cancer. And so this was inherited. If you looked at a pedigree, it was inherited as if the gene was a dominant mutation. And so what Ashley did is he looked at individual in these families that had this inherited mutation, and he could figure out which individuals carried the mutation. And then he did an age incidence plot. And so this is the incidence of colorectal cancer with individuals that have this inherited mutation at different ages. And this is extremely high. In this case, it's per million individuals. But this is 10,000. This is 1% here. This is 10%. So you see that 10% of individuals are getting colorectal cancer per year at age 30. So it's very high. And Ashley recognized, and I'm not going to explain this to you, but this is the case. He recognized that this curve, according to a particular theory of cancer incidence, should be higher, of course, because it's inherited. And the idea is that these individuals are inheriting a mutation, and these individuals, according to the theory, would have to acquire that mutation in their cells in their lifetime somatically. That is to say, in their own bodies, their cells would mutate as they're dividing, whereas these individuals have the mutation in all of their cells from birth. So of course the incidence is higher here because they have this mutation, and that the slope of this curve should be lower because the idea is that the number of different changes that are required determines the slope of this curve. And we'll possibly talk about that later. I'm just going to assert that. But in fact, this curve is exactly a slope of 1 less than this curve, which is what the theory suggests would be the case if being born with one mutation, one knockout gene, basically changes your rate of developing cancer by knocking out one of the normal protections in your body. Now later it was discovered that in fact these individuals who get cancer but don't inherit it almost always get the same mutation, but they get it in their tissues in a small piece of tissue in 1 or 2 cells initially, and then the tumor spreads from that tissue. And so this then really tied together a lot of aspects of how cancer develops genetically and the underlying causes of cancer, and also was exploiting this great regularity. So many of the interesting questions about cancer and causation come down to really parsing out what's really going on here. And that's something that I worked on a lot because I became very interested in cancer research over this. But today we're going to skip over that and use that just as the background. Um, and we're going to talk about is that these curves then for different cancers often have this shape. And one of the things you notice in this shape is that they tend to level off late in life. Now that doesn't mean that cancer is going down. What it means is that the rate's not increasing with age so much. So here about 1% of men are getting prostate cancer per year, but after age 80 they get it at about 1% per year. It's not going up anymore. It's a high rate, but it's no longer increasing. And you see the plateau for breast cancer as well. And this tendency for failures, breakdowns to decrease late in life is actually also known for mortality in general, and it's known as the puzzle. And this is a widely known puzzle. It's the puzzle of the late life mortality plateau. And here's the idea. Here's the death rate at different ages for humans. And then starting around age 90, the death rate, it goes up with age, but it starts to level off so that individuals, as they get older, they die, but they die at a faster rate, but that rate is starting to level off. Whereas if the rate of increase in the death with age was to continue as it was around age 80, it would go up like this, but actually it plateaus. And so the question is, why is there a late life mortality plateau in humans? And in fact, there's also one in Drosophila. So this is age in days for Drosophila. You grow up Drosophila, and the mortality rate goes up until they get old, and then it flattens out, and there's a late life mortality plateau in Drosophila. And so we see this in cancer, and in fact for automobiles. If you look at the age of automobiles that are around driving around, you see that as cars get older, they fail at a higher rate. But as cars get older, they fail at a high rate, but the rate is no longer increasing. There's a late life mortality plateau for automobiles. And so you see the same phenomenon in failure processes. And so that suggests that maybe there's some underlying general aspect of failure in the way we're taking measurements, which is giving us this regularity. And so anyway, there's this notion of regularity that we see in these observations. And this is a puzzle that hasn't really been solved. And when I was interested in cancer research, I spent a lot of time thinking about the things that are typical in cancer research. What's the relationship between the mechanisms and the genetics and incidence of cancer and so on. But I also became fascinated by these curves because there's just great regularity. And whenever you see something that's very regular and you don't understand why, I mean, that's sort of an instant trigger for a puzzle, to me anyway. And so then that made me interested in a very general way in regularities in data sets. And I started looking at regularities in other kinds of data and really paying attention to this. And so I ended up working on what I call the common patterns of nature. These are patterns in data which recur over and over again in seemingly different subjects. So here you have automobiles and cancer. You wouldn't normally think of that as going together. But what we have is a common pattern that is shared across different disciplines and different phenomena. And so I became fascinated by these common patterns because I felt that we have to understand these common patterns if we're going to understand the relationship between pattern and process in a general way. And so here's an example of a pattern, which is a common pattern in a totally different subject. This is a phenomenon that's common in ecology. These are tree sizes in forests. If you measure tree sizes in different forests around the world, you often get this pattern. If you plot the logarithm of the frequency of trees of a particular size, so you measure the size of trees by taking a tape measure and then taking the tape measure around the trunk of the tree or the stem of the tree. And then you measure how big that, how far around that is. That's called the diameter at breast height. So you stand there and at your breast you make a little circle around the tree and you say the circle has this circumference. So that's the classic measure of tree size. And then you see that, smallest trees are most frequent and biggest trees are rarest. So this is a frequency distribution plotted on a double log plot. Now when you do this for different forests around the world, you tend to see this pattern that you get a straight line on a double log plot, which is called the power law. but that for the very biggest trees, you get this decline. Now this is actually a small fraction of the total number of trees because we're doing log frequency. So this is just a couple percent of all of the trees here. But you see this tendency for decline. And this pattern is very regular. And this has led to a lot of theories about why trees have this pattern. And most of the theories are things like, well, you know, a tall tree dies and that causes a light gap. You have a light gap and then other trees will fill in the light gap and they'll start to grow up as small trees and fill in the light gap. And so you have a birth-death process that has to do with size, for example. And that can lead you to this pattern. But I look at this pattern and I think, yeah, you know, but automobiles and cancer have the same pattern. So is the answer here specific to trees and forest ecology? Or is the answer here that this is a generic pattern, just like automobiles and cancer, and if you try to explain it with a specific explanation, when the pattern is in fact generic, you're going to really miss out. So for example, here's a normal distribution or a Gaussian distribution. And I think most people get taught somewhere along the way in their education that when you see a normal distribution, that's the central limit theorem, and you expect that. And if you see a normal distribution of tree sizes, for example, you would not explain that by light gaps and something very complicated about trees. You would say that's just arises from measurements and averaging, and you don't give a special explanation for a normal distribution in terms of the specifics of the biology, because this is just a generic pattern. But then a guy like me, I wonder, what about that? Is that really special or is that generic? Maybe that's a normal distribution and the way people made measurements was just a transformation of the underlying measurements. If you take this and you transform the measurements in a particular way because you're measuring, instead of measuring the diameter around the tree, maybe you measure how much the trees weigh. And those are correlated measurements, but they're not the same measurements. It's a transformation. Maybe you're measuring some transformed measurement, and actually the trees are basically normally distributed, but what you're measuring is a transformation of that. If you transform that, maybe you get that. And in fact, a very simple transformation goes between those two distributions. And that, to me, is a very powerful, interesting question, because a great deal of science is looking at patterns like this in trees and in genomic data. This is the era of big data. We have huge data sets, and people are always finding patterns in their data and making explanations for their patterns in their data. But how often are they just seeing the central limit theorem and then telling a complicated story, which really has nothing to do with the data? This is one of the fundamental puzzles in modern science, because you can't really do modern science with big data unless you come to grips with this problem. But it doesn't get much attention. And the way I like to think about this is that think about those patterns I was showing you as forms. They're shapes, just like a snail shell is a shape. And if you think about the shape of a snail shell, we know that shells grow by adding material onto their leading lip, and that's what causes them to have this spiral, and they end up with what's called a logarithmic spiral. And that's a consequence of the mechanism of growth, of how they grow. So this is also a logarithmic spiral, but different in shape, but nonetheless has the underlying constraining form that arises from the pattern of growth. And so different shells look very different, but they're variations on a common theme. And so the question I like to ask is, when we see a pattern in data, when we see a pattern in shells, is it right to say that that pattern arises from a law of growth, or from natural selection that's caused a particular shape to happen because it's advantageous biologically? And the answer is always both. It's always that there are laws of physics which determine and constrain form, but that natural selection is always modulating form around the constrained patterns. And so when you look at this shape, it's a combination of natural selection, because this snail has a particular style of life, but it grows according to a logarithmic spiral. And so it's never right to look at a form and try to explain it in terms of a special explanation, like a particular adaptive story. And it's never right to look at it and explain it as purely an outcome of the laws of growth. It's always going to be both. Now I look at this form for the trees and I say, okay, it has something to do with trees, but it has something to do with the way the measurements are taken also. If you measure and aggregate the measurements in a particular way, that imposes strong constraints on the nature of the form of your data. Trees and their biology also impose things. So when we look at this curve, how much of that form is a consequence of the constraints of the way the measurements were taken? And how much of that curve comes from the special aspects of the biology? We don't know, but you have to ask that question. That question almost never gets asked when people look at big data sets. They tend to look at the data and then they tend to relate it to something very special because we love special explanations because that brings the biology in that we like. But if your data looked like that, you'd be more cautious. But how different is that from that? Maybe it's not that different. And in fact, when you look at those curves that are so smooth, is that caused by the genetics of cancer? Or is that caused by generic constraints that happen in something about failure processes that lead to that structure? And the answer is both. Because, well, the prostate and breast cancer look very different. And that's not a function of measurement, that's a biological consequence. At the same time, those curves are very similar in form. And that's probably a consequence of generic aspects of failure processes and measurement. Anyway, I'm fascinated by this particular subject, and I think it's not widely studied. And this is one of the great puzzles in modern biology to me, because so much of what happens in science comes down to understanding the balance between constraint, generic constraint, and special explanation. And this is really not dealt with very well. And I think especially in modern biology with big data, that there's a lot of open aspects here that really need work. So um, I wanted to emphasize that because I think this is we can talk about puzzles in this abstract way and, what we need to do to train ourselves, but also there's really some great scientific questions that we can look at of what are the puzzles of biology around us today that are really we need to solve to move ahead and really are the demanding things. What's the HIV of today? Of course, it's not exactly the same, but what I mean by that are what are the puzzles that are big, that are really out there that need work? And to me, that's one of them. And we've got 12 minutes. We can try the next one. I'll pause briefly, though. I have one more grand puzzle of the day to give you, but I'll pause for questions.
It strikes me that this special explanations being the thing that people want to talk about versus the general stuff. If you said the same thing to a physicist, usually they want the general things and not the special stuff.
Right.
So it may be like kind of confused that you said the special things were
Well,
the
that's
interesting
we can talk about that because you have training in physics. I've talked to a lot of physicists, and what you say is mostly right, but it's actually pretty subtle. Um, for example, all physicists are trained to know that invariances are the heart of modern physics. That's what Einstein actually did. Some people say that Einstein's greatest contribution to physics was to transform physics into the study of invariance. And most physicists would nod their heads and go, yeah, okay, yeah, that's true. Modern physics is that. But when you actually talk to physicists about where common patterns come from, they invoke Boltzmann's microcanonical ensemble, which is pre-Einstein and is not based at all on invariances. And it's because they actually like the special particular aspect of Boltzmann's microcanonical ensemble. And they have actually not internalized the invariances. So they know they're supposed to say invariance and generality is what it's all about. But as soon as they start to explain things, they invoke something which is not general at all. So there's actually a real tension in physics. Physicists are not as great as they think they are. In other words, I'm not picking on you, but there's a real interesting sociological phenomenon, which is that you're right, that physicists do tend to go for general explanations, and they do tend in that direction, and there's a lot of interesting things. But I've talked a lot to physicists and I've looked a lot at the relations between these things, and it's much more subtle than it appears at first glance, in ways that are really interesting, in ways that you need to understand if you want to do research at that interface. You have to figure out what's going on for yourself, because no one's going to tell you. In fact, what they tell you is actually very misleading. And so I've spent a lot of time looking at the history of physics and looking at what modern physics people trained in physics say, because there's a lot of physicists in biology now. This is a really interesting subject. And as I'm a theoretician. As a theoretician, this isn't just sociological curiosity. You have to understand these things if you're going to be a good theoretician, because you have to see where's the confusion. Where is it that people are saying things that's almost right but not right? And it often comes down to these really weird little historical things that you can trace back. In the cancer research, I had to write the history. I wrote a chapter on the history of cancer research because I couldn't figure out what was going on in cancer research until I studied the history of cancer research. And so I ended up writing a very comprehensive chapter on the history of cancer because there was no sorting out the problems until you got to the bottom of those things. This is very typical. So anyway, that's a good question, and I think these are non-trivial things that don't get the attention. And again, it's not. I'm very interested in the history of science and the sociology of science, but they're not just questions about history and sociology. They're questions that will really take you to the most interesting puzzles of the day in science. That's scientific puzzles that you need to face these things. Yeah, go ahead.
I had something that seemed interesting to me from one of your graphs that you had, where the automobile failure rate leveled off, as well as the human mortality rate. Those examples seem to me like they could be explained by simply filtering out those instances that are more likely to die based on some sort of external practice, like maybe somebody takes better care of their car, so once every other car has failed, they're still taking good care of it. But I don't understand how that sort of thing could also be linked to the cancer growth rate leveling off. I would think that the more cancer grows, the more it would be able to grow.
That's the heterogeneity explanation. So in cancer, what you would say is individuals have different genetic backgrounds, and what you're seeing are the individuals that are particularly susceptible are dying off, and then after all the really susceptible individuals die off early, what you're left with are those that are dying at a relatively slow rate, which is effectively the same thing that you said. And that's one of the common explanations for late life plateaus. It's not necessarily wrong. I actually do not think that that's what broadly explains what we see for a variety of reasons, but it's plausible. I think there's a more generic explanation, which is, I'll say it very fast. Most failures are the breakdown of multiple processes. You don't get cancer until a variety of checks and balances actually break down. We know this is true. Generally speaking, cars aren't considered broken until they break in a variety of ways. If the brakes don't work, you fix the brakes. But if the brakes don't work and the engine's not quite working and the transmission's shot, then you declare the car no good anymore. When you have a multi-step failure process, then you automatically get a late life mortality plateau. Since most failures are multi-step, then most failure processes will have a late life mortality plateau without invoking heterogeneity. Now, heterogeneity may be there, but you don't need it. If the process is inherently multi-step, which it almost always is, then you already have the plateau.
Okay, that's a really satisfying explanation.
Yeah, I actually feel it is. But actually, most people prefer the heterogeneity argument. Because I think the late life mortality the explanation I gave is a little bit more subtle mathematically to actually see how it works. So people have an easier time intuitively with the heterogeneity argument. And I think that's why it's more prevalent. It's not because the data are better. It's just a prevalence of an intuition thing. Um, so I
Steve,
won't.
can you give the reference for the mathematical argument? That would be very interesting.
Um, yeah, I mean, it's in my work, and if you look in my book, if you look on my website, there's a book called Dynamics of Cancer, and in that you'll see multi-stage models of failure. I don't talk very generally about failure there, but I do in other papers. Um, I can't think off the top of my head which paper would be. If that's not satisfactory, just send me an email. But it's pretty easy. You can just think of it as a gamma process. If you know enough
Hmm.
probability theory, a gamma process is usually taught as a multi-stage waiting time for n things to happen. How long is it before 6 buses go by?
Hmm.
How long is it before 6 things break down? So if you look at a gamma process and you make a log-log plot of the failure rate, you'll see it. The idea is very simple. If you're waiting for 6 things to fail, and then you look later and you look at a late time, then either every individual has failed or it has just one more thing it's waiting for to fail. If you have one more thing that you're waiting for to fail and you do a log-log plot of incidence versus time, you end up with a flat line because you're just
Mm.
waiting for one random event. So actually,
Yeah, yeah.
if you start with 6 things when you're born, and then you say, well, on average, when people are 60 years old, they won't have 6 things they're waiting for anymore.
I got it.
They've already gone through multiple steps. So it's an
Mm.
intrinsic to failure processes that you have a
Right.
demography of stages in the population that's heterogeneous, and you can actually calculate the distribution of individuals over states in terms of the number of failures that they've suffered. So for example, somebody like me, who's older than you are, I can tell you that in my colon, I have progressed through multiple stages of that failure process for certain, as have everybody else who's my age. You have progressed through probably, you may have made it through the first one, but maybe not. You're pretty young, but you'll get there. Um, it's inevitable, and so this is just an intrinsic aspect of the population demography of failure processes is that you have this. And so this connects up a huge number of things to, and so it's very interesting. You can't study cancer incidence without understanding this. But you can't study
And then,
cancer incidence without understanding the molecular biology of cancer either. I'm not saying that you know this, you don't need to know biology. I'm saying that you need to know both because they're both important in shaping the nature of failure, which is cancer onset is. But this is
so this is
rarely dealt with. So again, as I've emphasized, I think once you start thinking this way, you'll see this everywhere. And this is really one of the really great puzzles. I've written a lot about this general problem, not just about cancer. I'm totally, as you can tell, totally fascinated with it.
so to concisely say it back, we have a process. We can think of a system where the number of potential failures is decreasing over time. We can represent this as a gamma distribution, and when we look at the log-log presentation of it, we mistakenly get the signature of a power law. But the driver is that the number of failures is decreasing. Possible failures is decreasing over time, and if we work through the math, it just makes it look like this power law when in fact it isn't.
Yes, I would, if we were to have a little discussion, taking a mathematical view, what you said is 95% right. I would pick on a couple things that are not that are worth understanding, but you got it. You got the basic idea. And so once you're there, you can refine out the rest of it, or we can talk about it separately.
Mm-hmm.
But
Right.
but there's a couple technical things that are worth following up on. So that's the class for today. I didn't get to the last one, but I will start on the next class giving you a very brief introduction to the last issue because these are also some of the really great problems in modern science. And then we'll go on to um, overflow metabolism. So I expect, maybe 10 or 15 minutes for another puzzle. We'll do some overflow. Amber, were you wanting to ask a question? What I think what we'll do is it's 11:50. If you want to leave, just feel free. If you want to stay, we'll keep on talking for a few more minutes. So
I was just thinking about saving it till next week since we tend to go over what we just went over, right?
yeah, that's fine. Yeah, you'll
Okay.
have a question to start next class. That's fine.
Okay.
No problem. Yeah, if anybody wants to stay and ask a couple more questions, that's fine. But if you want to go ahead. So bye. We'll see you next week.
I think I have a boring question. What is the transform between the normal distribution and your power law that stops being a power law, that saturates?
Send me a chat and I'll send you the link to the reprint.
Sounds good.
It's called, I can't remember, I never remember the titles, invariances and power law, something. Anyway, I'll send it to you.
Sounds
because
good.
I actually analyzed that I used that as an example in a paper.
Okay. Perfect. Sounds good.
But also
Yeah.
more generally connected it up to power laws in general.
Okay.
Yeah, I'd be interested in that too. I'd initially thought that sometimes we can just mistake log normal distributions for power
Yeah.
law distributions.
But that's not the issue that I'm talking about. This is conceptually different. The paper's called Invariances in Power Law Size Distributions, and you can get it on my website. You're welcome to write to me, but if you just want to pull that down and have a look at it first, you may be able to get what you need out of it. Yeah.
That's great.
Cool. Thanks.
You're welcome. You're welcome.
One small question regarding actually some of what we talked about last week about overflow metabolism and biased gender ratios. I was wondering if in the cases where we observe that a biased gender ratio is present, or where overflow metabolism, that is to say, either a biased gender ratio or overflow metabolism is selected for, right? So we think that the conditions are conducive to that. Is the strategic structure that you're looking at there is a tragedy of the commons? Is that how you think of it?
Yes.
Yeah.
Yeah, it's interesting, but yeah, that's right. So if you trace the tragedy of the commons in the history of evolutionary biology, you can. But it took a long time to connect all those things together and relate them to the tragedy of the commons. But that's exactly how I think of it.
Yeah, no, it's great. It's always enjoyable seeing how often that pattern emerges.
It captures it captures the essential tension between individuals and groups, and that's an essential tension, and so it comes up over and over again. But it's amazing. You're right. It's amazing how that very simple, both mathematically and intuitively, how it captures so much. So yeah, I've long been fascinated by that. But it took me a really long time to recognize that the biased sex ratio problem was not kind of like the tragedy of the commons, but if you actually looked at the original equations, I developed some of the original equations for the tragedy of the commons in evolutionary analysis, and I worked a lot on sex ratios, but it was only much later that I kind of like, oh, it's exactly the same. You know, the males are, you know, could be thought of exactly as, not sort of like, but exactly formally equivalent to the same logic. So, but you're right. Conceptually, it's well understood that those are a very general way to look at these kinds of problems.
Yeah. There's one piece of the specific explanation that I'm aware I'm not grasping it because of my kind of lack of understanding of the logic of some of the biology. So and that is how males in those cases, like with the wasps, produce a greater rate of reproduction. I get that if they do, why that would be a competitive advantage, right? Because the trade-off is between having more offspring initially versus the ultimate number of offspring, where the idea is females set the limit of the ultimate number of offspring for the community, and males give an advantage to initial rate.
If you increase the number of males you have more grandchildren. And so the rate of reproduction is you need to count grandchildren, right?
Just like Fisher pointed out in his
Exactly.
original
So you need to count
observation.
grandchildren, and so reproductive rate is basically the number of since grandchildren is the unit of time, the number of grandchildren becomes a rate. So if you make more males, then you get more grandchildren than your competitors get. It's a relative rate. So increasing the number of males increases your relative rate of reproduction.
Yeah, forgive me. I'm slow in getting that one step. How does because what Fisher's argument is like, look, if the entire population, for example, is females, right, then having one male will net you more expected grandchildren.
Well, it's the same thing.
And
If the sex ratio is female biased in the local group, then if a mother makes more sons, she gets a relative reproductive advantage compared to the other female mothers in the group.
right, but it should go the other way, right? If there's a profusion of males,
Yeah, but there
then
isn't.
having one
But there
female.
isn't because we're starting from a. I took as a starting point that the sex ratio is female biased because the Nash equilibrium is a female biased sex ratio. So instead of looking at both sides, male bias and female bias, I'm taking the default as a strong female bias because that's where the equilibrium is. And then looking at variations around the equilibrium, you're still always looking at variations around a female bias sex ratio. So in all cases, when you deviate from the equilibrium by increasing the number of males, you increase reproductive rate. If you're at a 1-to-1 sex ratio and you increase the number of males you produce, you decrease your reproductive rate.
Right.
So it's distinctive because I'm taking as the framing a female bias sex ratio to begin with. That's all.
Okay. So these populations, for whatever reason, start with a female bias sex ratio,
Well,
and
it's not for
then
whatever reason. It's specifically for the reason of the local mate competition scenario we're studying. Because the ultimate equilibrium in the long run is going to favor a female bias sex ratio in this local mating scenario. So that's the frame of reference from which we're starting.
maybe I'll is there a nice article to spin myself up, because I can tell maybe there's a few assumptions that I'm missing at the same time, because I get Fisher's argument. Why
Well, you can try
we expect
Hamilton's
equal
original
sex
paper and
ratios.
see how you do with that.
That'd be great.
I mean, the size of the literature is vast, so you can try Stuart West's sex allocation book. If you want a purely mathematical stuff, you can look at my stuff. I probably have some papers where I talk about it in a more general way, but I have a lot of math arguments associated with it too.
Great. That'd be lovely. Thank you, Steve.
Yeah, just start and then send me a chat if you want to, or a text or something if you want to, after you look at a few things, if you want some more to read.
Great.
Okay?